Assalamu alaikum everyone, my name is Immad. I'm a third year medical student and today we'll be doing amino acid degradation. Uh, at any point if you have any doubts or whatever, you can text and I'll answer the question. And after the session, if you have any doubts, you can contact me on my email or my phone number. So let's get started. It shouldn't take too long. And I'll try to make it as simple as possible. So first of all, amino acids are degraded at, well, there's more than two sites, but the most important site is the liver. This is where the majority of amino acids are degraded. And then the second one is the muscle. The muscle is important because all of the branch chain amino acids, which include isoleucine, leucine, and valine, are degraded here. And this is important, and there's been MCQs on this in the past, so this is something that you must remember. Other than these two sites, there's a lot of other sites where minute amounts of amino acid are degraded, but it's not too important overall. These include the small intestine, the stomach, and other areas. So what happens is we have an amino acid and we remove the ammonia. And now we have a carbon skeleton when we remove the ammonia. What happens to the ammonia is not something that we care about in this lecture, but usually it's excreted or it may be incorporated into other compounds. So now we have the carbon skeleton. When this carbon skeleton is broken down, we can obtain one of 7 products. And I've listed those 7 products in the next slides, but let's forget about the products for now. There's 2 types of amino acids, either ketogenic or gluconeogenic. What does this mean? Ketogenic amino acids are those that upon the breakdown of their carbon skeleton, you have a particular end product. And a gluconeogenic amino acid is one in which upon breakdown of their carbon skeleton, you have a particular breakdown product. And those breakdown products are on the next slide, and we'll go to it now. So when we talk about gluconeogenic amino acids, that means when their carbon skeleton is broken down, you will have at least one of these compounds: pyruvate, oxaloacetate, fumarate, alpha-ketoglutarate, or succinyl coenzyme A. Now, when a gluconeogenic amino acid is broken down, it provides glucose, it's in the name, energy, and lipids to the body. Again, these 5 compounds, you must remember them because it gives you the definition of what a gluconeogenic amino acid is. Whereas a ketogenic amino acid, upon the breakdown of its carbon skeleton, you would have either acetyl coenzyme A or acetoacetyl coenzyme A. In fact, anything that comes after acetoacetyl coenzyme A, any derivative of it, also if the amino acid breaks down into that, it is also ketogenic. So for example, acetoacetate, although it's not listed here, but if a carbon skeleton is broken down into acetoacetate, that would also be a ketogenic amino acid because it's a derivative of acetoacetyl coenzyme A. And these obviously do not provide glucose to the body. That's why they're called ketogenic. They just provide energy and they can form lipids. So that's if that's clear, I'll move ahead because this forms the basis of this entire session. Now, which amino acids are gluconeogenic? So as you guys know, or maybe you don't know, there's 20 amino acids that humans must consume in a holistic diet. Out of the 20 amino acids, 18 are gluconeogenic. Now, since 18 of them are gluconeogenic, you don't need to remember their names. Just the 2 that aren't gluconeogenic are ketogenic. So everything except those 2 are gluconeogenic. And the 2 that are ketogenic, we'll do it on the next slide. But for now, just remember that majority of amino acids are gluconeogenic. Now, what I've written below, pyruvate, oxaloacetate, fumarate, succinyl coenzyme A, alpha-ketoglutarate. From the previous slides, you know that for an amino acid to be gluconeogenic, they must break down into one of these products. But how once they reach this stage of becoming this breakdown product, how do they form glucose? So pyruvate can enter gluconeogenesis. It's in the pathway. Oxaloacetate can enter gluconeogenesis. And the other three, they can be converted into oxaloacetate in the Krebs cycle. So the bottom three enter the Krebs cycle and then are converted into oxaloacetate. And then oxaloacetate is the one that makes new glucose. So they're indirectly supplying glucose, whereas the top two are directly supplying glucose. I hope that's clear. Now, the ketogenic amino acids are 2 out of 20, and these are the ones that are very important to remember. There are MCQs regarding this. So the first one is leucine, and the second one is lysine. So leucine and lysine are ketogenic. And how I remember this is just 2 L's for keto, because who wants to be on a diet? So what happens is, as I mentioned earlier, the carbon skeleton is broken down into acetyl coenzyme A or acetoacetyl coenzyme A or any of their derivatives, including acetoacetate. And both of these are part of ketone body synthesis or fatty acid synthesis somewhere in the cycle. Now, I have a question for you guys. Can amino acids be ketogenic and gluconeogenic at the same time? The answer to that question is yes. And the reasoning is because if you have an amino acid, if you have the carbon skeleton of an amino acid, and that carbon skeleton is broken down, it is possible for the breakdown products to include a ketogenic breakdown product and a gluconeogenic breakdown product at the same time. So if you have an amino acid that breaks down into acetyl CoA or acetoacetyl CoA, but then it also has pyruvate, oxaloacetate, fumarate, alpha ketoglutarate, one of these, then it's both ketogenic and gluconeogenic. And there are 5 amino acids that are both ketogenic and gluconeogenic at the same time. And again, this is something that is important to remember. And those 5 are phenylalanine, tyrosine, isoleucine, tryptophan, threonine. There's not much I can do to simplify this. You just need to remember the names. What's written after the names, what they break down into, is not too important. I just put it there for the sake of completion. But phenylalanine, tyrosine, isoleucine, tryptophan, threonine. These are the 5 ketogenic and gluconeogenic amino acids, so mixed. Now we move on to the essential amino acids. I'm sure you guys have done this in MOL 1. PVT Tim Hall is the mnemonic that people use. So phenylalanine, valine, tryptophan, threonine, isoleucine, methionine, histidine, arginine, leucine, and lysine. Now, arginine, if you notice, has a star next to it. And the reason for this is that it's not strictly essential. It's semi-essential because arginine is only essential when your body is undergoing periods of growth, such as in pregnancy, or you have a positive nitrogen balance. That's when arginine is essential. Otherwise, it's not essential. So it's classified as semi-essential, and that's why it has a star. But the mnemonic to remember this is a very famous one. It's PVT Tim Hall, private Tim Hall. And yeah, that's it. Not much to explain here. So we're done with the starting. This is the introduction. Now we're going to move to the degradation pathways. There's 6 pathways, and only 2 of them really are long. The rest of them are really simple. But let's get into them now, and we should be done soon, inshallah. The first one is degradation via removal of an amino group. So there are some amino acids, such as the ones listed on the screen, that can be converted into a useful product. A useful product in this case is anything that can enter the Krebs cycle and can be used to make glucose and energy. It can be converted into a useful product by just removing the amino group. Now, which ones are these? These are alanine, aspartate, glutamate, asparagine, glutamine. Again, the 5 reactions, you need to remember the starting amino acid, what they're broken down into, and the enzyme. So all 3 things are extremely important for you to remember. So alanine is broken down into pyruvate via ALT, which is alanine transferase, alanine amino transferase. And then aspartate is broken down into oxaloacetic acid via aspartate amino transferase. Glutamate is broken down into alpha ketoglutarate via an amino transferase as well. And then asparagine is broken down into aspartate via asparaginase enzyme. Glutamine is broken down into glutamate via glutaminase enzyme. So the last two are pretty self-explanatory, but basically, as you can see at the bottom of the screen, it says the ones in orange, which is the first three, these reactions involve transamination, which is the transfer of an amine group, and therefore aminotransferase enzymes are used. Now, the alanine aminotransferase, ALT, and AST, these are common enzymes that you must have heard of when you did GIT, because in liver damage they're elevated. So I'm sure you're familiar with these, even if you didn't know their full forms. But then you have glutamate becomes alpha ketoglutarate also via an aminotransferase. But the bottom two, asparagine and glutamine, these reactions involve deamination. So not a transfer of amine, it's a removal of the amine group. And these use specific enzymes that are named after the amino acids. Again, there's not much I can do to simplify this. You just need to remember the 5 amino acids that follow this degradation pathway, what they're degraded into, and the enzymes responsible. And again, there's been questions on this in the past. So yeah. Now, there is one clinical tie-in, which is asparaginase. So the enzyme asparaginase is important because it's used in leukemia and lymphoma, in the treatment of leukemia and lymphoma. And the reasoning for this is that normally, physiologically, human cells can produce asparagine. So it is not an essential amino acid. Again, if you guys don't know, essential amino acids are those that the human body cannot produce, and the humans are entirely dependent on their diet for consumption of these amino acids. And non-essential amino acids are those that can be produced in the body, so you don't need to ingest them. But asparagine is non-essential for normal cells. However, tumor cells cannot produce asparagine. So for a tumor cell, asparagine is essential. Now, we have asparagine in the body. What the asparaginase enzyme, when it's administered, what it does is that it breaks down the asparagine into aspartate. So there's less asparagine. So the tumor cells are deprived of asparagine. And since they can't make it, they're deprived, they don't have any asparagine, they can't grow like they normally would. So this leads to the death of tumor cells. And that's why asparaginase is used in leukemia and lymphoma. The specific type is L-asparaginase. Just keep that in mind, L-asparaginase. Now we're done with the first pathway, only 5 more to go. The second pathway involves phenylalanine and tyrosine only, not 5 like the previous one. So phenylalanine and tyrosine are actually related to each other because tyrosine is just a hydroxylated version of phenylalanine. So what happens is you have phenylalanine, and then phenylalanine is converted into tyrosine. But it's not that simple. There's obviously a cofactor. And this cofactor is called tetrahydrobiopterin. You also need oxygen for this reaction to occur. So basically, when you have tetrahydrobiopterin, then phenylalanine can be converted into tyrosine. And yes, please, you need to remember the name of the cofactor. Then tyrosine is broken down into fumarate, which is one of the products which makes gluconeogenic amino acid, and then acetoacetate, which was one of the ketogenic ones. So if you remember, phenylalanine and tyrosine, they're both a mixed amino acid. They're both gluconeogenic and ketogenic. And you can see that's represented in the reaction. So tyrosine, as you know, is a really important amino acid because it's responsible for the neurotransmitter, serotonin, and all of that. I don't think that's mentioned in this lecture in too much detail, but I think you will take it in a future lecture, or you might have already taken it actually. So yeah, tetrahydrobiopterin. Remember that. That's the most important thing on this slide. Now, it was mentioned slightly, so I put it in here. There's 2 metabolic defects that involve this pathway. The first one is phenylketonuria, and the second one is alkaptonuria. So phenylketonuria is a deficiency of the enzyme phenylalanine hydroxylase. This enzyme is responsible for the conversion of phenylalanine into tyrosine, like you saw on the previous slide. So what happens is when you don't have this enzyme, phenylalanine builds up in the blood. When it builds up in the blood, it can cause intellectual disabilities and a musty body odor. Alkaptonuria, on the other hand, is a deficiency of the enzyme homogentisate dioxygenase. And this affects the tyrosine degradation pathway. So it has nothing to do with phenylalanine. It's not involved. It's tyrosine. And what happens is you have buildup of homogentisic acid in the tissues. The main symptoms are darkening of the urine and joint pain, especially in the spine and knees, which appear at a younger age than expected. So yeah, those are the two metabolic diseases. They weren't mentioned in detail, but I've still covered them. But yeah, I think you will do them in a different lecture maybe. PKU and alkaptonuria. So we're done with two pathways, 4 to go. Serine, threonine, and glycine metabolism. So the first thing that you need to know is both serine and threonine are converted into glycine. So they don't have their own metabolism directly. They're first metabolized into glycine, and then glycine is broken down. So they're indirectly metabolized. So threonine, for it to become glycine, there's removal of an acetyl coenzyme A, and serine, for it to become glycine, there's removal of one carbon chain, just like one carbon. Then it becomes glycine, and I said glycine is the one that's actually broken down. So glycine can be broken down in two ways. There's a major pathway and a minor pathway. The major pathway is that it's converted into carbon dioxide and ammonium. And the minor pathway is that it's converted into glyoxylate and then oxalate, which is on the next slide. We'll go through it. But just remember, serine and threonine become glycine, and then glycine is broken down. And you need to remember what's removed from serine and threonine to make them glycine. Now, as I said, there's two pathways for glycine breakdown. The first one is it being converted into carbon dioxide and ammonium. It requires an enzyme called glycine cleaving enzyme. Again, this is important. Please remember this. And what this enzyme does is pretty, like, you can understand by the name. It removes one carbon from the glycine because it's a cleaving enzyme. The cofactors required are vitamin B6 and tetrahydrofolate. Again, very important for you to remember this. They do ask this, as stupid as it may seem, because it seems like it's not important, but this is something that they will ask you in the MCQs, the cofactors. Vitamin B6 and tetrahydrofolate are the cofactors for this reaction. And then remember the one carbon that was removed by the glycine cleaving enzyme? That is picked up by the tetrahydrofolate cofactor, and it forms methylene tetrahydrofolate. So basically, the THF, that was a cofactor, will pick up the carbon that was cleaved by the glycine cleaving enzyme, and it will transform into methylene THF. So you have methylene THF also formed in this reaction. And this is the major pathway for glycine breakdown. Then there's a minor pathway which involves glyoxylate. So what happens is glycine becomes glyoxylate, and then glyoxylate becomes oxalate. Now, this is important clinically because oxalate is responsible for the majority of kidney stones. Most of the kidney stones that you will see, 80%, are calcium oxalate. And therefore, when you see a patient with kidney stones, which is called nephrolithiasis, then for them, their glycine levels may be elevated. And the reason for this, obviously, is because if the glycine levels are elevated, then there'll be more glycine breakdown. And more glycine breakdown means more oxalate. More oxalate means a higher chance of precipitation. Higher chance of precipitation means kidney stones. So I hope that's clear. So we're done with 3 pathways. We just have 3 left. This pathway is probably the longest one. That's why I drew it out for you. But I've also written everything down on the next slide, so you don't need to worry about it. Methionine and cysteine metabolism. So basically what happens is you have this amino acid methionine, and it's converted into SAM. SAM is S-adenosyl methionine. And for this reaction, you require an ATP to be used. Once you have SAM, SAM is a very powerful molecule. And because it's powerful, it's unstable. So for it to become stable, it gives away its methyl group to any compound. In this, the example I gave was norepinephrine. So let's say SAM donates its methyl group to norepinephrine, and the enzyme that's responsible for catalyzing this reaction is methyltransferase. It's, again, self-explanatory because of the name. Methyltransferase transfers the methyl group. Then, once SAM removes its methyl group, it becomes SAH, or S-adenosyl homocysteine. S-adenosyl homocysteine is then hydrolyzed and broken down into 2 compounds, homocysteine and adenosine. Homocysteine is a harmful compound. It's involved in atherosclerosis, vascular diseases. It's implicated in, heart attacks due to ischemic reasons. So basically, homocysteine is not something that we want in our body. And when it is present, it's a red flag. So homocysteine can be broken down by 2 pathways again. The first pathway is homocysteine goes back and becomes methionine. But this pathway is not usually activated. This is only activated when the methionine levels in the body are low. So when the methionine levels are low, the body activates this pathway so you can have more methionine. And for this pathway, you require vitamin B12 as a cofactor, and you require donation of a methyl group, which should make sense to you because you remember Sam donated a methyl group, so now it needs it back if it wants to go back to being methionine. So two things for homocysteine to become methionine is a vitamin B12 and a methyl group. But more commonly, homocysteine becomes cysteine. And for this, it requires the cofactor vitamin B6, not B12, B6. Again, guys, the reason I'm stressing on this is because they will ask you for B12 and B6. They will ask you the difference. They will ask you different cofactors in different reactions. So vitamin B6 is the cofactor. It becomes cysteine. Cysteine, upon catabolism, will become pyruvate and sulfite. Sulfite is then oxidized within the body to become sulfate, and then sulfate is excreted in urine. So basically, all of the sulfate that you have in your urine is because of cysteine metabolism, more specifically cysteine catabolism. But that's it for methionine and cysteine metabolism. The next slide here, everything that I said is on this slide, so you guys can go through it. But again, I said everything. Every single thing on this slide has been said already. And then, again, this is the same thing. One thing that I would like to add is that cysteine this is something that they ask often, by the way, so please pay attention. Cysteine is made from the carbon skeleton of serine, but the sulfur group in the cysteine is donated to it by methionine. So a very common MCQ they ask is the sulfur group in cysteine is donated by which amino acid? And then the answer would be methionine. And the carbon skeleton is donated by serine. The rest of the stuff has been covered. So we're done with 4 pathways. We have one last pathway left, which is the histidine metabolism pathway. This is probably the most annoying one, but just stay with me. We're almost done. Histidine is converted into glutamate, which is then converted into alpha ketoglutarate. However, during the reaction from histidine to glutamate, we have a byproduct in between called N-formino-glutamate. So N-formino-glutamate, for it to be converted into glutamate, we need a cofactor, which we've already heard about before, tetrahydrofolate. So you can see the reaction on the bottom of the screen. Histidine becomes N-formino-glutamate. And then for N-formino-glutamate to become glutamate, we need a tetrahydrofolate. So what happens if we have a tetrahydrofolate deficiency is that we won't be able to go to the third step of the reaction. We're going to be stuck at step 2. There's going to be accumulation of N-formino-glutamate and its excretion in urine. So how is this important clinically? It's because to diagnose a deficiency of tetrahydrofolate, we give a pill of histidine. This is pure histidine. Histidine is given, and then the urine of the person is analyzed. So if the patient has excess N-formino-glutamate in the urine, that means the histidine metabolism couldn't be completed. And the reason for this is because there is a THF deficiency. So that's how this ties in clinically. And then the last step of the reaction, glutamate to alpha ketoglutarate, we need an aminotransferase enzyme to catalyze the reaction. Again, this one is a bit boring, I'm not going to lie, but you just need to remember the pathway. And again, the most important thing is N-formino-glutamate accumulation in the absence of tetrahydrofolate. Now, I've been saying tetrahydrofolate a lot, but I haven't explained what it is, so now I will. So it's a compound which involves a carbon group attached to nitrogen 5 or nitrogen 10. And the carbon group could be in any oxidation form, from formyl to methyl. So from 1 carbon to 3 carbons. That's why tetrahydrofolate is such a unique compound, because there's so many versions of it. So for example, the one that we read earlier was the tetrahydrofolate is required for the conversion of homocysteine into methionine, right? That's not exactly what we read, but if you remember, it says you need a CH3 or a methyl group for homocysteine to be converted into methionine. Now, where does this methyl group come from? It comes from THF, tetrahydrofolate, THF. And the version of THF that donates the methyl group to homocysteine for it to become methionine is N5, which means the carbon group is attached to the fifth nitrogen, methyl, so 3, and THF, N5-methyl THF. And this is the only time tetrahydrofolate acts as a methyl group donor. Again, this might seem out of nowhere, and there was a lot more than this in your slides. So I just picked this one because this is the most important one. Just remember that N5-methyl THF. Just remember it. You don't even need to understand. Just remember N5-methyl tetrahydrofolate is the only time THF acts as a methyl group donor. And the reaction that it donates this methyl group to is from homocysteine to methionine, which happens when methionine levels are low in the body. Now, what happens if you have a B12 deficiency or a folate deficiency? It can cause megaloblastic anemia. And why? It's because B12 and folate are required for purine and pyrimidine synthesis. This is one of your next lectures. And you'll see how both B12 and folate are involved in so many cofactors in purine and pyrimidine reactions. So if you don't have B12 and folate, you're going to have no synthesis of purine and pyrimidines. Purines and pyrimidines obviously make up DNA. So if you don't have purines and pyrimidines, then you can't divide the cell because you can't make another copy of the DNA. So the cell just keeps growing, which is why you have a macrocytic anemia. Macrocytic anemia, you guys already took heme, HLS. So you know it's the anemia where the MCV of the red blood cell is large, but the hemoglobin is low. So megaloblastic anemia is a type of macrocytic anemia caused by B12 and folate deficiency. Okay, so this is really the last one. There is one minor one after this, but it's not important. So this one is branch chain amino acid metabolism. I told you all the branch chain amino acids already because these are the ones that are metabolized in the muscles, not in the liver. Leucine, isoleucine, and valine. So a mnemonic, so the enzyme, and this is really important, and it's been tested multiple times before in the past. The enzyme involved in this BCAA metabolism is BCKDH, or branched chain keto acid dehydrogenase. This is really important, and this is one of the most important things in this lecture. So remember that the enzyme branched chain keto acid dehydrogenase, or in short, BCKDH, is part of the branch chain metabolism. Now, the important thing about this enzyme is that it requires 5 cofactors, 5. So a mnemonic to remember them is TLC for Nancy. And again, there's been questions on the cofactors, so don't think it's not important. It's very important. TLC for Nancy, the first cofactor is thiamine pyrophosphate, which comes from B1. There's lipoic acid, which is why you have L in the mnemonic. Then you have coenzyme A, which is why you have C. Coenzyme A comes from B5. 4 starts with F, so you have FADH2, which comes from B2. And then you have Nancy, which is why you have NADH, because they both start with N. And this comes from B3. So again, these 5 cofactors are really important to remember. Thiamine, lipoic acid, coenzyme A, FADH2, NADH. And these are very important for the branch chain metabolism. The enzyme, again, is BCKDH. Please don't forget. Now, this is the minor pathway I was talking about. It's not very important, but I still put it in here. Is the propionyl CoA pathway. So when you do fatty acid oxidation, or if you've already done it, you would have done the propionic acid pathway. So this pathway is the same as that. And there's 4 amino acids that also are part of this pathway. Another thing that you guys must remember is that it's not that one amino acid can only have one degradation pathway. Some of them have overlap. Like if you guys notice, methionine was also in the previous pathway, the methionine and cysteine pathway, but it can also be broken down this way. So there's multiple ways in which they can be broken down. But you just need to remember the ones that are given in this lecture. So a mnemonic for this is MIT Valorant. I don't know, I tried to make one up for you. MIT is pretty obvious, like college in the US. And then Valorant for valine, because VAL. Again, you need vitamin B12 as a cofactor. Because how this works is that methionine, isoleucine, threonine, and valine, which are the 4 amino acids, are converted into propionyl CoA, and then they go through a long step of reactions to form succinyl CoA. So vitamin B12 is the cofactor that's required for the reaction that because of which propionyl CoA becomes succinyl CoA. To be very specific, it's for the isomerization of methylmalonyl coenzyme A to succinyl coenzyme A. Again, you don't need to remember this. Just remember vitamin B12 is a cofactor for one of the intermediate reactions in the propionyl CoA pathway, B12. And remember that in B12 deficiency, if you have B12 deficiency, you may have methylmalonic acid accumulation, which could lead to neurological abnormalities. And if you're interested, I have written everything down, so you can just read it and understand. But just remember, B12 is a cofactor for one of the intermediate reactions. So we're done. Believe it or not, we're just going to do some questions, and then we're going to be over with this part of the lecture, but there's still a few more lectures, but I won't be giving them. Someone else will. But so let's do the questions. The first question is on the screen. Is there anyone in the meeting? Okay. So since there isn't, I'll just scroll to the next slide and you can see the answer. I'll also share the PowerPoint with you so you can see the answers later. So all of those questions, the four previously, were in the doctor's lecture. So again, please be familiar with them because she might bring one of them in your final. The next four questions are from exam committee. They're from past questions and practice questions. That's it, guys, for my part of the lecture. I try to make it as short and as easy as possible. Usually, the questions from this lecture are straightforward. They'll just ask you. It's just remembering stuff. So I try to make it easy for you with mnemonics and with tables. So that's it. Please scan the QR code. And thank you. Now my colleague Razan will take over and you're going to do the rest of the lectures with her. So good luck and I hope you all do well in your MOL final, inshallah.
Okay, salam alaikum everyone. My name is Razan Elhassan. I'm a second year medical student. Inshallah, for today, I'll be explaining metabolism of phenylalanine and tyrosine and the other amino acids metabolism. Feel free to contact me if you have any questions, either if you joined right now or later on my phone number or either the email. Okay, so here, these are the objectives I'm going to cover, and at the end, we're going to solve some questions. So as just now, my colleague Immad, he explained the essential and non-essential amino acids. The essential ones that we need to get them from our diet, and the non-essential that they like de novo, it's de novo synthesis. We're going to synthesize them in our body. And adding one thing to what Ahmad mentioned regarding arginine, why is it semi-essential? Because in addition to these 2 points, what he mentioned is that it's required for one of the enzymes in urea cycle, which is CP1, CPS1. Okay, so that's why it's considered semi-essential. And here's the mnemonic for the essential amino acids, PVT, Tim Hall. Make sure you know them and don't mix the tryptophan and threonine with tyrosine, because sometimes it can happen, especially in MCQs where they get you, for instance, tryptophan and tyrosine, and you just remember it starts with D, so don't mix that. So again, previously in enzymes lecture, I spoke about the inborn error of metabolism, where we said it's due to mutation in an enzyme that leads to accumulation of one of the substrates, and the product. So by that, we don't have the product. Okay. So regarding the screening, also we mentioned that we screen for that 2 to 3 days after birth, because at that period of time the infant will still have the normal level of enzymes because they got it from. So here we go, phenylalanine and tyrosine. So from these two amino acids, so there are three important metabolic disorders that I'm going to speak about: the phenylketonuria, albinism, and alkaptonuria. So first thing, phenylalanine. Phenylalanine is going to be converted into tyrosine by the enzyme phenylalanine hydroxylase. And this enzyme, to be activated, we need BH4. So if we don't have BH4, this enzyme won't work and we don't have tyrosine. By that also we can have phenylketonuria. Okay, so how we can get BH4? From BH2 in the presence of NADH and the enzyme dihydropyridine reductase, we can get BH4, and a side product is NAD. Okay, so make sure that you know that phenylalanine hydroxylase enzyme need BH4. So here we go, the first disorder, phenylketonuria. It's deficiency in the enzyme phenylalanine hydroxylase. So either it's the enzyme itself is deficient, or we don't have the cofactor, which is BH4. So let's say that this enzyme is not there. Having the enzyme not there, phenylalanine would accumulate, and that accumulation would lead to deficiency in tyrosine. And tyrosine is very important for the other neurotransmitters and hormones like like serotonin. Serotonin is considered neurotransmitter and can be also a hormone. So phenylketonuria would be, phenylalanine would be converted into phenyl pyruvate, phenyllactate, and phenylacetate. So if a patient So as I mentioned, deficiency in phenylalanine hydroxylase activity would lead to accumulation of phenylalanine, and by that phenylalanine is going to be converted into phenylketone. So the patient would present with phenylpyruvate, phenylacetate, and phenylacetate and phenylacetate. Sorry, this is a piece, I think. Yeah, phenylacetate. These both would be present in urine. So by that, you can confirm that this is phenylketonuria. We have two types of deficiencies, like the classical one where the enzyme is deficient, and the other one, which is rare to be present, like the deficiency of BH4, which is the cofactor for activating this enzyme. So what are the signs and symptoms that this patient could present with? This is a keyword like in the questions, like this patient presents with musty smell in breath and sweat or urine. And also this patient could present alongside this one, like the musty smell in breath, is like they have pale skin, fair hair, blue eyes. And why is that? Because they lack tyrosine, and tyrosine is needed for melanin production. So this is another keyword in the question. But if you saw just that they have pale skin and fair hair and blue eyes, this means that this is albinism, which I'm going to talk about later. But this must be in the question, the musty smell in breath. So how do we treat this? Like the dietary modification, like we restrict phenylalanine, give them specific type of food, and most important thing that we don't give them aspartame. Aspartame is found in bubble gums and sparkling drinks and these things, so we shouldn't give them aspartame. And at this point, we need to give them tyrosine since the body cannot make it. Again, here, as I said, the BH4 deficiency, same thing. Like BH4 is not there, so the enzyme won't work. Why is not there BH4? Because the enzyme dihydropyridine reductase is not there. So we cannot synthesize it. And again, the treatment, same thing. We just supplement giving BH4, levodopa, levodopa and carbidopa, which we need them for dopamine, and hydroxytryptophan, which we need it for serotonin. And here also the dopamine, when there's deficiency in dopamine, we have increase in prolactin, as also dopamine is known as the inhibitory prolactin inhibiting factor. So if we have a lot of dopamine, there will be a decrease in prolactin. So at this point here, we don't have dopamine. So that's why prolactin would be increased. Again, here, this is the screening, as I mentioned earlier. And here, this is a summary for phenylketonuria. It's a good summary from Dr. Hannah's slides, so make sure you go over it. Then here we have tyrosine metabolism. I just wrote all the steps together. From tyrosine, we get DOPA, and here the rate-limiting enzyme in this reactions or pathway is tyrosine hydroxylase. It's the rate-limiting enzyme, and it's activated by BH4. Then DOPA is going to be converted into dopamine, and here we have DOPA decarboxylase, where we remove carbon dioxide. And here we need a B6, and you need to know also that this B6 is called pyridoxal phosphate, as we were asked about it, like in the choices, the answer was pyridoxal phosphate instead of B6. Dopamine, again, is going to be converted into norepinephrine by the enzyme dopamine beta hydroxylase. Here we need also a cofactor, which is vitamin C, to activate this enzyme. Then finally, norepinephrine is going to be converted into epinephrine. And here at this stage, we need SAM. This enzyme, I don't think they're going to ask you about, which is phenylethylamine N-methyl transferase. But you need to know that we need SAM at this step, okay? And here and here we have one of the mnemonics by one of our colleagues in our batch. Big black van to kidnap Sam, whereas big is B4, black B6, van is vitamin C, and here Sam is for Sam at the last step. So this will help you to remember these cofactors which is needed for each of the enzymes here. Then here we have SAM. So what's SAM? SAM is considered a cofactor that we needed to donate methyl group. So how do we get SAM? We need to have methionine and ATP, and by that we can get SAM. So here we need methionine. So how do we get methionine? This part of getting methionine will be explained here and again in the second lecture too, the whole pathway. But here, as briefly, folate, vitamin B9, from it we can get N5-methyl THF, okay, tetrahydrofolate. Then this methyl tetrahydrofolate, in addition to homocysteine, and in the presence of B12, which is cobalamin vitamin, we get methionine. So this is the way we get methionine. Also, again, here as an important side note, SAM, when it donates its methyl group, it's also give you homocysteine and adenosine. Okay, so here in the second step, we have clinical significance. DOPA converted into dopamine in the presence of DOPA decarboxylase. Okay, here the clinical significance is Parkinson's disease. Parkinson's disease, where there is decrease in dopamine production in the brain, and we need to treat that. So how could we treat it? We treat it by L-DOPA and carbidopa. So here there is also an important thing that we were asked about last year, that why do we give carbidopa? We could just give levodopa, and levodopa is going to be converted into dopamine. So we don't give levodopa alone. We give carbidopa alongside it because we have also dopa decarboxylase preferably, not only centrally in the central nervous system. So we give carbidopa. Carbidopa is going to inhibit dopa decarboxylase, allowing the conversion of L-dopa in the central nervous system instead of peripheral nervous system. Also, levodopa, if it's converted peripherally, it would lead to several side effects, such as nausea, vomiting, and other side effects too. Also, one of the presentation of Parkinson's disease, if it was mentioned in the question, mask-like face, pill-like tremor, and also resting tremor and shuffling gait, and also they have rigidity, which is called also like cogwheel rigidity. Then we have thyroxine and melanin. We get also thyroxine from tyrosine. So tyrosine, in addition to iodine, you're going to get thyroxine, which is T4. Melanin, as we said, that it's pigment for skin, hair, and eyes. We get melanin from melanocytes, and melanin is a polymer of tyrosine. So you have tyrosine. They get into like becoming cycles using the enzyme or they cyclase by the enzyme tyrosinase, and they become dopaquinone. This dopaquinone, like it's multiple structures of tyrosine that gives us melanin. Don't confuse melanin and melatonin together because this can happen too. So here, as I said, if there's deficiency in tyrosine, we're going to get oculocutaneous albinism. Oculo is the eyes, and cutaneous is the skin. Albinism is like there's no color or this white color. So we have two types of deficiencies. Again, the classical one, which is tyrosinase, the deficiency in the enzyme itself, or in the tyrosine transporter. If not mistaken, I'm not sure about it, but I think the tyrosine transporter is the dopaquinone. I could double check this and let you know about it. So this is the most common one, the tyrosinase. And decrease in tyrosine, yeah, and this would be the presentation. So pale skin, blonde hair, and blue eyes. And this would increase the risk of sunburns and skin cancers. Okay, here we come to tyrosine breakdown. We have the tyrosine here. It's already in the body. It's going to be degraded into homogenistic acid. Then this homogenistic acid is going to be degraded into fumarate and acetoacetate using the enzyme homogenistic acid oxidase. So let's say now we have deficiency in this enzyme. This metabolite would accumulate and lead to alkaptonuria. Alkaptonuria is deficiency in the enzyme homogenistic acid, leading to polymerization. So this metabolite, homogenistic acid, is going to accumulate and polymerize, leading to dark pigment. So if they told you a patient presents with, first thing, they would tell you that the patient, we took a urine sample in the beginning, like first hours, it presented, like it looks normal. Then we left it a couple of hours and it turns to black color or it turns dark. So note out that this would be alkaptonuria. Also, this pigment or the homogenistic acid would also accumulate in the cartilage and also in the spine or the spinal cord. So this is a classical X-ray finding, classification of the intervertebral disc. Yeah, and urine discoloration, as I mentioned here. So these diseases are very high yield. Make sure you know them. Of course, there will be like at least one of these diseases would be asked. Yeah, so here the diagnosis and treatment. As I mentioned, elevated homogenistic acid in urine or in plasma. Treatment is just dietary restriction of tyrosine and phenylalanine. So this part, the catecholamines, I don't think so. We will be asked about it a lot, but just know it that catecholamines are degraded by MOL or this monoamine oxidase or اللي هو catechol O methyltransferase. So these two are used to let me explain it here in the picture, are going to degrade either dopamine, norepinephrine, and epinephrine. So it doesn't matter by which one it started, either by MAO or by COMT. So if it's degraded first by MAO, it's going to be degraded again by COMT, then it's going to give you homovanillic acid. Again, same thing if it's COMT or MAO. Here for norepinephrine, same thing. It doesn't matter by COMT or MAO. The end product would be vanilla mandelic acid. So if a patient presented with, let's say, with adrenal tumor that it's secreting a lot of these catecholamines, we measure the urine levels for this vanilla mandelic acid. If it's increased levels, this means this patient have pheochromocytoma. Recently, they're not using this one or this metabolite. They're using normetanephrine and metanephrine. So either one of these three, or actually these both are used together, and this is used a lot. So either one of them could lead to diagnosis of pheochromocytoma. So here's the same thing, pheochromocytoma. Last thing here, we have the pharmacology. If a patient has depression, there will be decrease in the levels of neurotransmitters like dopamine, norepinephrine, serotonin. All of these neurotransmitters would be decreased. So if we want to increase them, we give them MAO inhibitors, where there would be no degradation of these neurotransmitters, and by that their levels would increase in the brain, and we can decrease the progression of this depression. Finally, here is a summary of this lecture. You can go over it. Even it includes this maple syrup urine disease, which will be explained in the next lecture, inshallah, now. And this is related to the fatty acid synthesis, if not mistaken. So here are the questions. If someone would like to answer that. So we can go over it. Like here, they say that this patient, a child from Nigeria, is evaluated for developmental delay. His coloring seemed much lighter than that of his family background, and his physician orders blood amino acid test that demonstrate elevated phenylalanine. Here, from this part of the question, you can see that you have decrease in. Okay, someone answered B. Yeah, deficiency of tyrosine and melanin. So yeah, as I mentioned, decrease in these two, or sorry, as the who answered. Yeah. So as he mentioned, it's deficiency of tyrosine and melanin due to his presentation. So this patient came in with decreased color, and he's different from his other family members. This indicates tyrosine and melanin deficiency. Okay. Next question. C. Phenylalanine hydroxylase deficiency. So the key word here would be that the patient has chronic seizure. Yeah, they have phenylpyruvic and phenyl acetic acid. And as we mentioned, that phenylalanine is going to, since it's accumulating, it's going to be converted into phenylketone. Next question. Okay, no worries. So it says here genetic engineering has been used to create a strain of mice that are unable to synthesize dopamine. These mice can survive embryonic development only if if they are rescued by provision of an external source of dopamine. Which of the following genes would be most likely target for inactivation to produce such dopamine deficient mice? So since this mice don't have dopamine, means that it's tyrosine hydroxylase. So it's basically here it's asking about the enzyme. Wait, sorry for that. Yeah. Yeah. So here there's dopamine deficiency, and from the tyrosine metabolism pathway, the rate limiting enzyme for that step is tyrosine hydroxylase. So if you don't have that enzyme, you cannot get dopamine. Hope it's clear right now. Next question. Let's see, yeah, homogenistic acid oxidase. So yeah, here it says because it's wait, sorry, yeah, it's not homogenistic acid. Read the question again. It says like white hair. Let's see, yeah, dihydrofolate reductase. Oh, oops, sorry, I just mixed. It's not C, it's D tyrosinase. It's the enzyme itself is not there because why? Because the patient here presented with white hair, red appearing eye colors. I think you cannot see that, but basically, having this presentation, as I mentioned earlier, it's sorry about that. Yeah, it's deficiency in tyrosinase. So tyrosine is going to be converted into dopaquinone, and dopaquinone will be converted into melanin. That's why here it's the answer. Okay, yeah, no, I got you. It's not, it's the, yeah. So I think this is it was the last question. Now we go with the other amino acid metabolism. Again, here, these are the objectives, and at the end, we're going to do some questions. So yeah, first thing we're going to start with tryptophan, niacin, serotonin, and melatonin. So in the reaction, we have tryptophan. Tryptophan is going to be converted into 5-hydroxytryptophan, and then this metabolite or intermediate is going to give us serotonin. So here the enzyme is tryptophan hydroxylase. Again, here this enzyme needs BH4. Okay. Tryptophan gives us serotonin. And then again, we take this serotonin here, and this serotonin is going to be converted into melatonin. This melatonin is going to regulate your sleep and awake cycle, which is different than melanin. Again, don't mix them. This is for sleep and wake cycle, and melanin is for the color, basically skin color, eye color, and these things. So make sure you don't mix them. Again, from tryptophan, you can get niacin, which is vitamin B3, in the presence of vitamin B6 pyridoxal phosphate. Then niacin is going to give you NADH and NADPH. So as I'm watching your recording, your doctor gave a question that which of the following amino acids can give it was neurotransmitter and a vitamin. So the answer was tryptophan. So such questions can be asked. Then here we go to serotonin degradation. So we said that just now. Patients with depression, they need to be give these MOL inhibitors so we can increase the levels of dopamine, serotonin, and norepinephrine. So these MOL inhibitors can also, instead of them, we can give SSRIs to also do the same job, but they only act on serotonin. So they just only increase serotonin by inhibiting its reuptake. So instead, they prefer to use MOUs because they act like broadly. They prevent the reuptake of dopamine, serotonin, norepinephrine, and these things. Okay. So here, histidine to histamine. Histidine is converted into histamine by histidine decarboxylase. And here also we need vitamin B6. So generally know that amino acids reaction, we need vitamin B6 to be present. And also, since it's decarboxylation, it's going to give carbon dioxide as a byproduct. Okay, so yeah, this slide is just, I think, an extra. So here, glycine. From glycine, we can get heme. So what's heme itself? So heme is just a porphyrin ring, and here in the middle there should be an iron atom. But we need carbon atoms, and we need nitrogen. So from where we can get them? We get them from glycine and succinyl-CoA. So this is the source of the nitrogen and carbon that we have in heme structure. Then we have arginine. Arginine can give us keratinine, urea, and nitric oxide. So if they asked you, what are the three things that arginine can provide? They are either keratinine, urea, or nitric oxide. So keratinine is synthesized in the liver and in kidneys. So glycine, we have glycine, and arginine, we get keratin. In the presence of SAM. SAM here donates methyl group. Then we get this keratin here, and we want to convert it into keratin phosphate. So which enzyme do we use? We use keratin kinase. This keratin phosphate is going to be, or it's going to cyclase a reaction that it's just going to be like, I think, in a cycle shape, and it's going to give us keratinine. This keratinine is excreted in urine, and it's used for kidney function tests. So also, one of the things I think you should be took it in renal, like if a patient came in and he's athlete, and the doctor measured his levels of keratinine, and they were like, these levels were very high, maybe the doctor will send this patient directly to dialysis. But the issue is that here, this could not be a true finding, or like right finding. Why? Because all this athlete is playing whatever in gym and these things. So the muscles also produce keratinine. So it's not due to, here the increase in keratinine is not due to kidney failure, but it's due to muscle production. So you need to differentiate between these things too. Here are also the products of arginine, and just put them in case you want to see them all together here. Here we have glutamate. From glutamate, we can get GABA. So glutamate is an excitatory neurotransmitter in the brain, and GABA is the inhibitor neurotransmitter in the brain. And here the enzyme that we use it is glutamate decarboxylase in the presence of vitamin B6. Here, the branched chain amino acid. So these branched chain amino acid, they're metabolized in the muscle cell. So as my colleague earlier mentioned, know that the only amino acids that are metabolized in the muscle cells are the branched chain amino acids. By which enzyme are they metabolized? By branched chain alpha keto acid dehydrogenase. And here are the cofactors that are needed: thymine pyrophosphate, lipoic acid, coenzyme A, FADH2, NADH, and there is a mnemonic. I think Dr. Hannah also mentioned it, the lovely coenzymes for nerds. This would help you to remember them, because they asked about that previously, that which cofactors, which of the following cofactors is needed for branched chain amino acid dehydrogenase enzyme, or alpha keto acid dehydrogenase enzyme. So here is the degradation pathway: valine, transamination, remove the amino acid, the amino group, and then decarboxylation, and then dehydrogenation, and then you can get succinyl-CoA. And same thing for isoleucine and leucine. Then here we have one of the disorders related to the branched chain amino acids. So if we have deficiency in the enzyme branched chain alpha keto acid dehydrogenase, these amino acids would accumulate, leading to what's called maple syrup urine disease. So here you have one of the findings is urine have a sweet smell. So which of the amino acids that leads to this is the isoleucine. And also we have neurotoxicity. It's also due to accumulation of leucine. So these things could be your keywords in the question. Again, here the diagnosis is just the elevated valine and leucine, isoleucine in urine. So how do you treat them? Just dietary restriction and monitor their plasma concentration, and also supplement them by thiamine. Because thiamine is one of the cofactors that's needed for this enzyme. So it could be deficient, this thiamine. Then we have homocystinuria, also one of the high-yield diseases. So here's the pathway that I mentioned that I'm going to go over it again. So first thing, we have folate, which is B9. From folate, you get N5-methyl tetrahydrofolate. You take this N5-methyl tetrahydrofolate, in addition to homocysteine, you're going to get methionine and tetrahydrofolate in the presence of the enzyme methionine synthase and the vitamin B12 cobalamin. Okay? Then methionine, in the addition of, to ATP, you're going to get SAM. Then after that, SAM is going to donate its methyl group and give adenosine and give homocysteine again. Okay, so if you want cysteine, you're going to take homocysteine and add to it serine. So it could be asked which of the following amino acids could be added to homocysteine in order to generate cysteine. So it's serine. And the enzyme is cystathionine synthase in the presence also of B6. So just a fun fact, if the enzyme ends in synthase, that means it doesn't need ATP, but if it ends in synthetase, that means here we require ATP. So here, if you have elevated levels of homocysteine, it could be due to folate or B12 and B6 deficiency. And this also is associated with increased risk of cardiovascular diseases. But let's say someone said, okay, let's restrict this homocysteine or decrease its levels so we can decrease the level of or decrease the risk of cardiovascular disease. In some studies, they said that this lowering this level of homocysteine does not lower the risk of cardiovascular diseases. Then here, the homocystinuria, the disorder itself. So it's due to defect in homocysteine metabolism. It's due to defect in the enzyme itself, which is cystathionine synthase. So the signs, or the patient would present with lens dislocation, long limb and chest deformity, as you can see here in this diagram. Also mental retardation, blood clots, and early atherosclerosis. I'm not sure if you took Marfan syndrome, but you can distinguish homocystinuria because Marfan syndrome patients also present with the same signs, but the only thing that differentiates them is the blood clotting or blood clots. So having blood clots would indicate homocystinuria. So here, again, this is the enzyme, which is cystathionine beta synthase. How do you treat them? They should avoid methionine, and this is repeated in questions a lot, where they should avoid methionine, and you should increase cysteine. Why is that? Because you don't have methionine, so you cannot get cysteine at the end. So even if you got if you have here methionine is going to be converted into homocysteine. The homocysteine is going to give you cysteine. So since the enzyme is deficient, you cannot give or you cannot have cysteine made in the body. Also, you can supplement them with vitamin B6, and these patients showed that they respond better than those who are not getting vitamin B6 alongside the treatment. Other less common causes is methionine synthase deficiency, the enzyme which is at the beginning here. Yeah, here, deficiency in this enzyme could be one of the causes, but it's less common. And also defect in metabolism of folate and vitamin B12, which is like MTHFR mutation, which is this enzyme here, which is the enzyme required for folate conversion into N5-methyl tetrahydrofolate. And yeah, that's it for this lecture. Here we have now the questions. You can go ahead and answer them. Yes, A, just the tongue. So the patient presented with ectopic lens and it needs to be replaced. Okay, good job. Next. B, yes, maple syrup urine disease because it's due to branch chain amino acid. Next question. Is E, yes, good job. And here, finally, I think last question. Good. I think it's the same question. Oh yeah, same question. It was the last question. Thanks for listening and being here. If you have any question, feel free to text me. Please scan the QR code for feedback to further improve my thought.